our next speaker. So we've got Dr. Marike Geldenhys, that's it. I did try and say it earlier and then I struggled just now. But yes, thank you very much for coming. She's from the University of Pretoria, and she's going to talk about understanding coronavirus maintenance and excretion dynamics within bat populations. A very, very important topic in this current day and age. So please carry on. Thank you.
Hello everyone, thank you for joining us today. My name is Marike and I am a postdoctoral researcher at the Biosurveillance and Ecology of Emerging Zoonosis Research Group at the University of Pretoria, working under Professor Vanda Makotter. My research focus and the focus of today's talk is on coronavirus surveillance and essentially all that entails. So we perform coronavirus surveillance in the natural hosts of these viruses, which typically are bats and small non-virulent mammals like rodents. And we do this to get a better understanding of these viruses and how they emerge. As a brief background, coronaviruses are known to infect people, causing common colds, and then I think everyone is familiar with COVID, SARS-2, which caused such an unprecedented pandemic. In the last 21 years, 3 new human-infecting coronaviruses have emerged: SARS coronavirus, SARS-2, and MERS coronavirus, the latter originating from the Arabian Peninsula in 2012. If anyone has any questions about these viruses, we can discuss them at length during the question section. Coronaviruses also infect mammals and birds, which can be fairly severe diseases. And then coronaviruses affect livestock animals quite severely and are responsible for a large proportion of annual morbidity and mortality amongst farmed animals. There has even been a newly emerging, reoccurring coronavirus affecting pig populations in Asia. So in total, 4 new emergent coronaviruses in 21 years. I just want to highlight that taxonomically, there are 2 genera of coronaviruses mostly associated with mammals: the alpha and the beta coronaviruses. As an example, all 3 of the human viruses on the slide are beta coronaviruses. And then the pig virus, called SADS, is an alpha coronavirus. There are also known human alpha coronaviruses like NL63 and 229E. We know from significant global surveillance activities amongst wildlife and domestic animals that bats host a wide range of genetically diverse coronaviruses. These viruses are also widespread amongst these bat populations and have been found on all continents where bats also occur, even the islands. A number of these bat coronaviruses have been determined highly similar to known coronavirus species occurring in other animals, with the suggestion that there is a long history of crossing the species barrier. So due to this possible animal involvement in the emergence of new viruses, investigating coronaviruses amongst livestock and wildlife are important for their potential to spill over and cause disease in new hosts. A lot of surveillance research has been performed and has drastically expanded the known genetic diversity of coronaviruses. However, we still have very limited understanding of how these viruses are maintained in their host populations and which mechanisms are involved in spillover of bat coronaviruses. To answer these questions, one needs lots of good quality data and ideally to investigate the bats, their viruses, and their environment together and over a long time span. We need to improve our understanding of coronavirus infection amongst individual bats, how populations of various sizes and demographic makeup affect the circulation of these viruses, and whether these factors are universal for all host species and different types of coronaviruses, as most results have been broadly generalized. Animals in the shared habitat may also host their own cohort of coronavirus species that may be capable of recombination when combined with bat coronaviruses via spillover activities, or how anthropogenic activities in the area may result in increased or even decreased risk of spillover opportunities or even stress on animals. We can then bring all of these factors together to develop a more accurate picture of what is happening and then express that as possible risk to spillover or emergence. There have only been a few studies that have investigated the maintenance of bat coronaviruses, though the last year and a half has seen a sharp increase in the literature regarding coronavirus excretion dynamics from various wild bat populations with different ecological niches, helping to determine which aspects are important to consider when assessing risks and where interventions can be applied. The reason why so few studies could do this research is because it is very labor, resource, and cost intensive. It requires frequently visiting a site on a predetermined schedule and collecting enough data for thorough statistical analysis as well. And so few groups are in a position to be able to do this. So most of the literature so far would agree that the higher proportion of bats shedding coronaviruses can be identified amongst young bats, the sub but also the immature bats, around the time that they are being weaned and becoming independent, suggesting that these bats are becoming susceptible to infection, possibly due to a loss of maternally derived antibody protection. So beyond this, the roles of bat cohorts in various reproductive life stages and their involvement in the maintenance of coronavirus species and populations are a little less clear-cut. What is important to highlight is that we have very poor understanding of how long immunity lasts and when bats can become reinfected. To investigate this, you would need to be able to recapture bats within a reasonable time frame, and due to logistical difficulties involved, only a few studies have been able to report results on this from wild-caught bat populations, especially with strong statistical support. So for our study, we wanted to investigate the coronaviruses present within a maternity colony of Rosettes aegyptiacus in the Limpopo province of South Africa, where there is an interface between bats, other wildlife, and domestic animals near the roosting sites, as well as people living and farming in the area. Rosettes aegyptiacus is a cave-dwelling fruit bat called the Egyptian fruit bat and is somewhat widespread throughout Africa. From the literature, we know that there is a good chance of detecting some coronaviruses from a population of this species, but we wish to determine how the infection dynamics changed over time, if peaks of excretion could be pinpointed, and if shedding occurred at only certain times of the year or were constantly excreted. We then aim to also characterize the species we identify and define the host or temporal factors associated with the circulation of the virus within the host population. We performed monthly sampling at this maternity roost for many years now, though we've analyzed 2 years worth of data, so we are still currently sampling at the site. We collect mainly 2 sample types for this specific study: rectal swabs taken from bats collected in hop traps at the cave entrance. We also record morphological measurements, their age, sex, and current reproductive stage, so thus we had demographic data for the bats. And then we also collect fecal samples non-invasively from inside the cave. This was to expand our sample sizes per month and essentially to obtain a larger sample volume for genetic characterization. Because we couldn't link fecal swabs in the cave to specific hosts, we pooled these together as 3 to reduce the costs. So we tested the extracted samples for coronavirus nucleic acids with PCRs. We then sequenced these positives and evaluated the positive proportions over time in a generalized linear model, as well as a multinomial regression GAN in R. We used a combination of amplicon-based Sanger and Illumina sequencing for sequencing the full genomes and the spike genes. As I mentioned, the surveillance we're doing is still ongoing, though we have analyzed the first 2 years of data in the meantime. Overall, we had 201 coronavirus nucleic acid detections from 1,430 gastrointestinal samples, with a comparable positivity between the fecal samples that were pools and the rectal swabs, which was overall 13.2%. Surprisingly, we detected 3 different coronavirus lineages circulating in the population at the same time. If we look at these viruses, phylogenetically, there is 1 alpha coronavirus shown in blue and 2 beta coronaviruses shown in red and green. You will note that the 2 beta coronaviruses are not identical, with some genetic differences to each other. The alpha coronavirus is very different to previously reported species and likely represents a new species. The dominant beta coronavirus, which represents the majority of the positives, shown here in red, is very similar to a coronavirus species called HKU9 and occurs in fruit bats like rosettas from Asia and various other African countries, sharing about 90 to 99% similarity in the short region from the surveillance. The last lineage, which is in green, was very rarely detected in our study and only shares between 72 to 74% nucleotide identity to this HKU9 related diversity. So what does this mean and why is this relevant? The coronavirus I just mentioned, HKU9, is a member of the Nobicavirus subgenus within the beta coronavirus genus. And Nobicaviruses generally have only been found in fruit bats. These viruses are not currently linked to human or animal disease, but have been recognized as potential candidates for the emergence or establishment in new host species by several independent research groups, as well as spillover risk ranking tools. The reason for this is that these viruses are widespread and occur wherever old world fruit bats occur in Africa, Asia, and Australia. Nobicaviruses are quite diverse and have speciated into a number of different species and variants, suggesting that they are undergoing frequent mutation and recombination. There are 3 recognized species. I'm just going to highlight. HKU9 is from Rosetta leishmanii in Asia. And then in Africa, there's a species described from Eidolon helvum, as well as widespread diversity related to HKU9, with reports from various countries that are highlighted in green on the map. So when you look at the coronavirus genome, it is divided into non-structural genes, which is about 2/3 of the genome, with the last third being the structural genes. So everything towards the back of the genome is more interesting, as it is the structural proteins that one needs to investigate for possible cross-protection or pathogenesis. The spike gene, of course, being responsible for receptor recognition and attachment to host cells. Open reading frame 1AB is used for species demarcation, as there are currently 5 domains required for this that occur in open reading frame 1A and several in 1B. There are various ways to characterize the genomes of the viruses detected, from primer walking and high-throughput sequencing of longer amplicons to unbiased approaches where the input DNA or RNA need to meet specific concentration requirements. Not to get into too much detail or overcomplicate things, we are busy with the alpha coronavirus genome. It was very diverse and has been a little complicated. We used an unbiased deep sequencing approach to recover some of the genome, as shown in the figure, with the covered regions, and we were successful in recovering 68% of the genome. So we're currently busy with some Sanger sequencing and primer walking. We also recovered the complete genomes of both the HKU9 related lineages, 4 in total, as well as the novel beta coronavirus. Between this novel beta coronavirus and this HKU9 diversity, they only share about 64% identity, suggesting that this novel virus is actually a new species that occurs in rosettas in Africa. Then the 4 HKU9 related viruses we sequenced represent new strains of this HKU9 diversity, of which there are only 5 other African Nobicavirus genomes to compare these to. And because it is too expensive to sequence the genome of every PCR positive sample, we started to sequence the spike genes, so far of 41 of this HKU9 diversity, to identify any mutations or changes in these genes over time. Though we have only analyzed the spike genes of 8 so far, because even this is no small task, because the gene is 4,000 base pair long. So you need an approach like next generation sequencing, or you will be doing a lot of Sanger sequencing. We were quite surprised by the sequence variation amongst the spikes. They were not all the same, and overall shared between 92 to 99% amino acid similarity, with less similarity to the spike gene of HKU9. The colored percentages show the similarity within a spike cluster, and the gray shows the similarities amongst the sequences detected in the South African bats. In this tree, we see the Kenyan Rosetta sequence grouping with other sequences. This is expected because they're all from rosettas. Surprisingly, there's a high similarity to Eidolon helvum HKU9 related spike genes, though this is a completely different host genus. And all of these are most similar than when compared to HKU9 spike. We cannot yet determine if certain variants have become extinct and others more dominant, as we need to sequence more spike genes. And even among the receptor binding domains, there's a lot of differences between the variants we identified, ranging from 88 to 93% similarities only. The receptor targeted by HKU9 is also currently unknown. So we plotted the coronavirus excretion over time to see if there are seasonal trends or those associated with reproductive stages. We at first used GLMs and then moved all the parameters into a multinomial regression GAM model, which you can see plotted here. So these graphs indicate positivity over time. The solid line for rectal swabs and the fecal collected in the cave displayed by the dashed lines. We are still building and refining the model to see what are important parameters to consider, but you can see clear peaks at the start of the year, even if the positivity in 2018 is higher than that in 2019. If we go back and consider what is happening in the population, there is only one birth pulse among rosettes in southern Africa. Bats accumulate in the colony in spring, starting in September. Mating still occurs at that time. Births begin towards the end of October and November, with pups becoming independent immature bats from February to April, when most of the colony then leaves in May, with a small population remaining behind. You can see that there is almost constant excretion from the population, though there are significantly larger peaks at the start of each year, that coinciding with the influx of the young weaning bats becoming independent. We can also see that the dominant HQ9-related beta coronavirus accounting for the majority of the trends we see overall, with a lesser extent the new alpha coronavirus. If we then look at the demographics of the bats found to be excreting coronaviruses versus the total sample per month, we see that the young bats were indeed the ones driving the excretion peaks during certain times of year, with a positive correlation between coronavirus detection and the abundance of subadults. This being true for all lineages except the rarely identified beta coronavirus, as there are too few data points to make statistical inferences. Even between the sexes, there were no statistical significance in infection between males and females, especially when added to the multinomial regression analysis. Then lastly, all bats that we sample also receive a numerical tattoo so that we can track the positivity of recaptures. Tracking recaptures sound easier than it actually is, as we recapture a very small number of bats. Only about 75 bats, which is about 10 to 11% from the 710 individuals tested, represent recaptures. Of these, only 19 bats were positive for coronavirus RNA, and 10 of these bats only could be used to determine possible viral clearance estimates, as they were positive as their first capture. There was also one bat positive over 2 consecutive months, though for a beta and then an alpha coronavirus, which may suggest clearance of one infection does not confer immunity to a different type of coronavirus. This is based on one data point though. Overall, it suggests that infection duration likely doesn't last more than a month, with clearance that may occur within 3-4 weeks. This is also in agreement with some other studies from insectivorous bats from Asia and Australia, though due to the low numbers of recaptures, it's difficult to make more concrete conclusions at this stage. And we can't yet comment on duration of immunity or when bats may or may not be reinfected with coronaviruses. So we identified the viruses in our population. We also characterized these viruses recognizing new species and expanding strains of known species. From the longitudinal surveillance, we identified that age is an important contributor to the maintenance and circulation of coronaviruses in this rosetta's population. The idea being that the young bats are becoming independent as they are being weaned and losing their maternally derived antibody protection, if such a thing even exists. And from a few other members of the population that are carrying these viruses, these young bats are then becoming infected and passing it along to each other. Thus, the virus has become amplified within the population. The riskiest time of year, if you want to label it as such, for possible spillover to occur is at the start of the year. The coronaviruses are being shed throughout, and more than that, these frequent changes amongst the spike genes within the receptor binding domain warrant further investigation, so that we can determine if there are possible adaptational changes that may be relevant to spillovers. Next steps will be to expand on serology to determine the issues around immunity, expand surveillance to other terrestrial animals in the area, as well as there's a new project that started this year investigating human interactions with bats and wildlife in the area that may help us identify risk behavior. And of course, research like this requires a huge team. So to all the students and staff at the Bees Research Group, thank you and well done. Then of course, we've also received fantastic funding for Prof. Makoto's research, especially from the NRF, the CDC and DITRA, and then also the PRF grant awarded to myself. Lastly, thank you to everyone for joining us today.
Thank you very much, Marike. Are you around to answer questions? There you are.
Yes.
So I don't see any questions in the chat. I suppose the question that I have is more to do with what happens next. Obviously, you've got this fantastic comprehensive research that explains when shedding is most likely, when your high risk times are, and obviously the next steps are looking at human behavior. But what happens in terms of those cave sites that maybe require monitoring of people going in and out at those times? Do you have any ideas of what could happen in the future to help with that?
Yeah. So when we started our research, they actually used the cave for religious ceremonies. And after a few years, when we've realized some of the viruses that are circulating in the Rosetta's population specifically, we told them to maybe reduce that or to stop, and we wanted to also identify, we wanted to see if we could accommodate them as well. If they had to use the site for religious reasons, could we tell them not specifically during certain times of the year? That's also why we wanted to know, what kind of excretion is occurring. Is it peaks at certain times of the year, or is it constant? Because I was speaking about coronaviruses today, but we also do surveillance for other pathogens at the specific site that includes paramyxoviruses and filoviruses. So filoviruses, of course, being the ones most important that you would want to prevent people from getting exposures to, like Marburg virus, for example. So we did highlight to the locals that there are times of the year when it is worse versus other times of the year. So if they could, reduce the times that they go into the cave, but they actually have spoken to each other and they actually don't go into the cave that frequently anymore. Even though we do put up camera traps in the aim of catching animals, we have also caught people going into the caves, even though they say they don't. Yeah.
Thank you very much. Yeah, there's a cave that's actually got quite a lot of activity near us in Lusaka that we are a little bit concerned about, and the University of Zambia has been monitoring for a long time. So maybe I can put you in touch with them and see how they may be able to collaborate in future.
Yes, we actually had a workshop in April last year, and I can't remember the lady's name now, but she was also at the University of Zambia, and she told us about Rosetta's they are also sampling. So yes, definitely, let's all share details and protocols and things like that. Very helpful. Yeah.
Absolutely. So I think you're talking about Katendi. So she's fantastic, and definitely would be great to get you back in touch. So I'll definitely do that. Thank you. In the interest of time, we will have to move on. But if anybody has any more questions, I can see a couple more coming up there in the chat. Please may you answer these in the chat, Marike, if that's okay.